In this second slide series, we will explore the structures and functions of genomes, including the genomes of eukaryotes, prokaryotes, and viruses. We also will explore the human genome in depth and learn how changes in the structure or number of chromosomes in the human genome lead to certain genetic disorders. And so in our first series of lectures, you should certainly now have a good idea of the structure, the physical structure of DNA itself. You should now understand replication. You should know that each time a cell goes to divide, it needs to replicate its DNA. And you should also know about transcription and translation, which is the process by which we start out with DNA and the genes encoded within that DNA to make an mRNA message copy that then gets translated to a protein. If we go, though, and look at the simple structure again as a review, if you recall, DNA is bound, or I should say, in a structure called a chromosome, wound up into a tight structure called the chromosome. Each chromosome of a living cell is a DNA double helix. The atoms that make up the DNA are organized into two primary molecular components. If you remember, they were the sugars with the phosphate groups and the bases attached. And it was those bases attached to the sugars with the phosphates that gave the different nucleotides. There were 4 bases for DNA: adenine, cytosine, guanine, and thymine, abbreviated A, C, G, and T. Now, each chromosome, as I had mentioned, is a double helix, and then the chromosomes within the nuclei typically are linear. And so there really is so much DNA that you need these second higher order structures in order for all of that DNA to simply fit within the nucleus. Embedded within the DNA sequence, if you recall, of each chromosome are the organism's genes. And so the chromosome can be thought of as the DNA scaffolding within which the genes reside. And an organism's set of chromosomes is called its genome. Genes are interspersed unevenly along the lengths of most eukaryotic chromosomes. Across the human genome, for example, we find very gene-rich regions of the chromosomes, and then in other regions there are very gene-poor regions, meaning there's just a lot of nucleotides, but they don't necessarily encode for anything. The genomes of most eukaryotes are fairly large and often complex, and in addition to the genes themselves, most eukaryotic genomes also contain a variety of non-coding structural and regulatory elements, as well as the introns. Some of the genetic material serves as a fossil record, if you will, or a history book written in biological terms and handed down from generation to generation. And what do I mean by that is as we evolve, our genomes tend to pick up junk, and we hold on to some of that junk over time, and it kind of gives us a look into how the genome's evolving and what sort of things have made their way into the genome over time. For example, we often find a lot of retrovirus or retroviral elements in our genome. And so at some point in time, these viruses or pieces of these viruses have become embedded in our genome and just sort of stick around over the course of time. Now let's talk a little bit about overlapping genes. This is maybe a bit technical, but bear with me here, because the normal concept that people think of is that genes are lined up one next to each other along the span of the nucleotides that make up the chromosome. But that's not always the case. The genes within an organism's genome are generally not evenly distributed along the length of that organism's DNA, and particularly for eukaryotes. In eukaryotes, as I had mentioned, there's very gene-rich and gene-poor regions within the genome, where in the gene-poor regions we find a lot of non-coding spacer regions that span the distance between the very gene-rich areas. Now, even within the gene-rich areas, despite all the DNA that we have, we find that there are overlapping genes, and this is actually quite common. Overlapping genes are different genes whose nucleotide sequences overlap along a segment of the DNA. The nucleotide sequence of the overlapping genes is read by RNA polymerase in 2 or more reading frames or from opposite strands of the DNA molecule. Thereby, you can encode two different proteins within the same segment of DNA. Now, one gene leads to one protein, right? Or one mRNA in eukaryotes leads to one protein molecule. But in order to generate that first mRNA, we can find that there are overlapping genes within the DNA segments. Overlapping genes are found throughout nature. This is not unique to eukaryotes. Although prokaryotes and viruses tend to have their genes immediately adjacent to one another, we do find overlapping genes in bacteria and viruses as well. Now let's talk a little bit about viral genomes, and then we'll talk a little bit about prokaryotic genomes, and then we'll focus on the eukaryotic genomes. But this is just to give you a little broader perspective of genes and genomes in general. Viruses, just as a quick review, are not cells. In essence, these are little DNA or genome capsules. They are genetic material encased in a protein coat called a viral capsid, and that's like a round ball shell that protects the genomic material inside. Some complex viruses also surround themselves with a viral envelope or membrane that's made up of lipids and glycoproteins, and a good example of that is the HIV virus. The genetic and biological characteristics of viruses varies widely. There's many different types of genomes, viral genomes. Some are single-stranded, some are double-stranded. Viral genomes can be made of DNA. There are such things as RNA viruses. So it's a much more diverse group of genomes found in viruses than, say, what you would find in a eukaryote. Like bacterial genomes, the genomes of viruses tend to be very small. They typically do not contain introns. However, despite their small size, the genomes of viruses can be quite complex in that the genes can overlap or lie immediately adjacent to one another, and ultimately you can get interesting splicing of the genes that ultimately results in several different types of proteins. Moving along to prokaryotic genomes, there are also, again, a little bit of diversity amongst the bacterial genomes. Prokaryotic genomes are made of DNA. We do not see RNA genomes in the prokaryotes, but we do see some differences in terms of the organization of the genomes. Prokaryotic or bacterial chromosomes can be circular, and this is different from what we see in the humans where they have all linear chromosomes. But in bacteria, we find both circular or linear chromosomes. And prokaryotic cells do not contain organelles, just as a reminder to sort of help you keep things straight. Now, the genome size can also vary widely. Some prokaryotes have very small genomes, a few 100,000 base pairs, or several, or can be very large genomes, as large as several million base pairs. It just depends on the particular organism that you're looking at. Now, if we move on to the eukaryotic genomes, and this is the one group we've been focusing on, like the extensive diversity in life forms within the eukaryotic domain, the genomes of eukaryotes also vary greatly in size and composition. And to give you an example, if we look at the yeast Saccharomyces cerevisiae, the genomes are around 12.5 million base pairs in size. Now, this may sound big, but if we compare that to a particular fern genome, Psilotum nudum genome is about 250 billion base pairs in size, so that's quite a bit different. Further, most eukaryotic genomes encode complex genetic elements that preserve and maintain the structure of the genome and regulate transcription or expression of the eukaryotic genes. And so as we move from the viruses through the prokaryotes to the eukaryotes, you see not only the size of the genome expanding, but you also see the complexity of regulation of the genes increasing as well. Again, as a review, the genomes of eukaryotes contain those regions known as introns. Introns are those non-coding segments of DNA. They're of variable size. They separate the coding segments, or the exons. And ultimately, during mRNA processing, the introns are spliced out or cut out and removed from the RNA molecules to form mature mRNA. That's then 5' cap, 3' poly A tail prior to exiting the nucleus for translation on the ribosome. In addition, as I had already mentioned, the genetic material of eukaryotes is organized into one or more linear structures called chromosomes. Now, we do not see circular chromosomes amongst the eukaryotes. And in the next couple of slides now, we're going to focus specifically on the human genome. And this is where the video focused on, and this is where the rest of our talk will center on. So the human genome, the haploid human genome, consists of 23 different chromosomes. The diploid human genome consists of 23 pairs of chromosomes. When we're talking about the haploid human genome, we're talking about the chromosomes found in the egg and the sperm. And in those sex organ cells, that's where you're only going to find 23 chromosomes per cell. Once you have merging of the sperm and the egg, it's now considered a diploid cell. And the diploid human genome consists of 23 pairs of chromosomes. 23 chromosomes came from mom, 23 chromosomes came from dad. One of each pair of chromosomes is inherited from each parent. As I had said, one from mom, one from dad. The first 22 pairs of human chromosomes are called autosomes. The 23rd pair of human chromosomes determines the gender. And so human females carry 2 X chromosomes, one inherited from the mother, one inherited from the father. Human males carry 1 X chromosome and 1 Y chromosome. Males inherit their X chromosome from their mothers, and their Y chromosome comes from their fathers. In addition to the nucleus, you should be aware that the mitochondria of human cells also carries 1 or more copies of a small circular DNA molecule. And the mitochondrial chromosomes are inherited through the egg. Sperm do not contribute mitochondria to embryos. So as a result, genetic diseases caused by mutations in genes on the mitochondrial chromosomes are passed through the maternal lineage, from mothers to all their children. Children of fathers affected by mitochondrial transmitted genetic disorders are not at risk for inheriting the diseases. However, because mitochondria also import proteins encoded by nuclear genes, the inheritance of diseases of the mitochondria do not always follow this exact pattern. Moving along with the human genome, the haploid human genome, as we had said, the 23 chromosomes, contains approximately 3 billion base pairs of DNA. Therefore, the diploid human genome, the 23 pairs of chromosomes, contains approximately 6 billion base pairs of DNA. To put it in perspective, only approximately 1.5 to 3% of the human genome actually encodes for proteins. It is believed that much of the rest of the human genome serves some sort of regulatory and or structural functions. The exact number of genes carried by humans is not yet known, but it is likely to be somewhere between 35,000 and 100,000, and that's still a pretty big target. Humans are more than 99% identical to each other at the DNA level, and only identical twins possess identical genomic DNA. It is that less than 1% variability in our DNA that makes each of us unique. And in recent years, the variability in human DNA has been used as a fossil record to study human history and trace the migration of humans out of Africa as we spread it across and populated the earth. What we have learned from our DNA is clear. Every human on the planet today is related to every other human. We are all descended from the same trunk of the human family tree. And in essence, then that makes us all brothers and sisters, separated only by time and geography. If the DNA contained within a single human cell were laid out lengthwise, with each chromosome arranged end to end, the DNA would be 5.8 feet or almost 2 meters long. However, it would be very, very thin, only 24 angstroms wide, and it would take 26 million DNA molecules arranged side by side to measure 1 inch wide. So we're talking a very tiny, thin molecule, but extremely long. So in order to fit inside the microscopic nucleus of a human cell, the length of the DNA must be packaged very precisely. And in this first level of packaging, the DNA double helix is wound very tightly around proteins called histones. Histones are made up of 8 component proteins, and they're often referred to as histone octamers. The packaging of DNA around histones gives DNA sort of a bead on a string appearance. And then histone packaged DNA results in a 10 nanometer fiber. Remember, the native DNA double helix is only 24 angstroms wide. So this is 100 angstroms or 10 nanometer fiber is about 4 times wider and already quite a bit shorter than the native double helix would tend to be. The histone packaged DNA then is further wound into 30 nanometer fibers, and then these fibers are then packaged into loops of DNA believed to be anchored to the cytoskeleton within the nucleus of the cell. DNA that is then completely packaged is referred to as the chromatin. Now, as I mentioned, there's a lot of regions of DNA, particularly in the human genome, that do not encode for proteins. So what are they there for? Well, scientists are still working to figure this out, but there are several clues and several things that we have already uncovered. So when we talk about these regions, we often think about regulatory mechanisms. And so the expression of genes within the human genome is controlled by a number of different mechanisms. Sometimes you want a gene turned on so that the proteins can be made, and at other times the cell doesn't need those particular proteins, and so a gene will be turned off. The cell has to maintain energy efficiency at all times. And so there's a number of things called promoters, enhancers, and silencers. And these are segments of DNA that regulate the rate and amount of transcription or expression of the nearby genes. Now, if you remember, transcription is the process of synthesizing the mRNA as a messenger in order to implement the instructions encoded with DNA. And so what are promoters? Promoters are generally regions located immediately upstream from the first exon of a gene. Enhancers, and what you'll get is you'll get the RNA polymerase binds to these promoter regions and then can initiate transcription. The sequence of the DNA can affect how strong or weak a promoter is, and by that I mean how well the RNA polymerase may or may not bind to that region of the DNA. Enhancers are genetic elements that increase transcription of associated genes, and therefore silencers are genetic elements that suppress transcription of associated genes. And these enhancers and silencers can be located some distance away from a promoter, and they may be upstream or downstream of the promoter on which they act. And so all three of these things work together or in concert to determine how often an mRNA should be transcribed off of a particular DNA sequence. In addition, there's 2 other mechanisms, and I'm only going to touch on these quickly, methylation and imprinting. Both of these, again, are regulatory mechanisms or ways to control whether genes are expressed and at what level they're expressed at. In addition, 2 other types of genetic regulation involve alternative splicing and also for RNA stability. And so some mRNAs are more stable than others. The ones that are decaying quickly obviously aren't going to make as much protein. They're not going to be translated as well. And in addition, while we talk about one gene, one mRNA, one gene hypothesis, we do find, though, that there are what we call alternative splicing. And meaning you have here, as you see in this transcript, the 4 different exons, and it just depends on how that mRNA gets cut up, and can change or alter, I should say, the type of protein that's made. Now, one thing that students often find interesting is this idea of regulation of the human genome through X inactivation. And I bring this up because it's often not known that, in essence, in females, one of the X chromosomes is silenced. So during X inactivation, what happens is most genes on one of the X chromosomes of a female are inactivated in a mechanism that provides dosage compensation to equalize the level of gene expression of most X-linked genes in females to that of males who only have one X chromosome. So you're kind of trying to even things out here. This process, often called lionization, results in the formation of a bar body. And so if you were to look under a microscope, you see this highly compacted, inactive X chromosome. And you see in this particular photograph, the bar bodies are shown by arrows. The form of DNA in the inactive X chromosome is called heterochromatin, and it's wound up very tightly, and the transcription machinery cannot get to the particular genes, and so in essence you only have gene expression off of one of the X chromosomes in females. Now, in human females, one X chromosome or the other is inactivated in each cell early in development. And it's a random process, if you will, as to which one of the X's gets inactivated. So as a result, some human female cells have one chromosome active, while the rest of the cells may have the other X chromosome active. And as I had mentioned, it's typically a random process or nearly random, meaning that half of a female's cells express the genes of one X chromosome, and the other half generally express genes from the other X chromosome. Now, in some females, though, X inactivation is not random, and it's skewed. And in cases of non-random or skewed X inactivation, all or most of the cells of a female express X-linked genes from a single chromosome. In reality, non-random X inactivation may be less representative of the process of X inactivation than of reduced survival of cells with a particular X chromosome inactivated, making it appear that X inactivation was non-random. In some cases, females who carry X-linked genetic diseases can be affected by those diseases if X inactivation is not random. For example, if a structural abnormality disrupts the pattern of X inactivation, then most or all of a female's cells will express genes from a single X chromosome. If there are genes that are disrupted by the structural abnormality and those genes cause a disorder, the female can then show symptoms of the disorder. And for many X-linked disorders, female will show mild to moderate symptoms. And lastly, I'll end by looking at a few different karyotypes. The image in this slide in which you see the condensed chromosome is a karyotype, and this shows the karyotype from a normal human male. If you look, you'll note the presence of one X and one Y chromosome. Karyotyping is a laboratory technique which allows for the microscopic visualization of the number and gross structure of the chromosomes within a cell. And now this karyotype would be abbreviated 46, XY, 46 for the number of chromosomes and XY for the sex chromosome makeup. If this karyotype were from a normal female, there would be 2 X chromosomes and no Y chromosomes. A normal female would be designated 46, XX. Now in this slide, there are 2 separate questions for you to look at. Number 1, first determine is the individual a male or a female? And secondly, can you identify the chromosome abnormality in this karyotype? Now, if you look at the sex chromosomes, you'll see that this individual is a female. Note the 2 X chromosomes and the lack of a Y chromosome. This individual carries 3 copies of chromosome 21, though. And this chromosomal aneuploidy is called trisomy 21 syndrome, or you've probably heard of Down syndrome. Down syndrome is caused by the presence of an extra copy of chromosome 21. And so this karyotype would be abbreviated 47, XX plus 21. Individuals with Down syndrome can have a variety of abnormalities, including mental retardations, speckling of the irises of the eyes, poor muscle tone, characteristic appearance of the face, some cardiac abnormalities, and loose skin in the back of the neck. Individuals with Down syndrome are also prone to leukemia, premature aging, and Alzheimer's disease. It affects approximately 1 in 800 live births. Approximately 75% of Down syndrome fetuses spontaneously abort during gestation. And of babies born with Down syndrome, most carry an extra chromosome 21. And lastly, can you figure out again, is this individual a male or a female? And can you identify the abnormality in the karyotype? In this case, you should have picked out that the individual is again a female, noting 2 X chromosomes and the lack of a Y chromosome. Of note, this individual carries 3 copies of chromosome 13. And this is a chromosomal aneuploidy called trisomy 13 syndrome or Patau syndrome. The karyotype would be abbreviated 47, XX plus 13. In this case, babies with trisomy 13 can have a variety of different congenital abnormalities, including incomplete development of the forebrain, mental retardation, a sloping forehead, cleft lip, skin abnormalities, etc. Trisomy 13 occurs in approximately 1 of every 5,000 live births. More than 95% of fetuses that are trisomic for chromosome 13 spontaneously abort during gestation. Most babies with trisomy 13 that are born alive will unfortunately die within the first few months of life. So in summary, genomes contain the genetic information of organisms. I hope we've made that clear over our past few lectures. Large genomes undergo extensive packaging to facilitate their enclosure within the cells. There's a variety of different ways or mechanisms, I should say, for regulating expression of genes within an organism's genome. And we also covered different types of karyotyping in the human genome.